Thank you. Thank you, everybody. Thank you to you all for being here. I was very honored and very flattered to receive this invitation because it means a lot to me that I get an opportunity to tell you all about the work that we're doing at the Institute of Cell Biology. I will not have time to go into the details of what we are attempting to do in the newly established group, the medical parasitology group, but I will have some time to tell you a little bit about what we're doing with the Multidisciplinary Center for Infectious Diseases. So this is going to be a two-part talk. And as I was mentioning earlier, my biggest fear is to be running overtime because there's so much that I want to tell you all. So please make sure that I know when it is that I have to stop.
So you have about 50 minutes and then we'll...
I have about 50 minutes.
Go on the big shepherd's talk.
I think I'm going to be able to stay within time, but do give me the heads up if you feel like this is taking too long. So protein trafficking in Giardia Lamblia. This is the protist that made me see the light back in 2010 when I started at the University of Zurich with Adrian Hell. Adrian Hell had just transitioned from a very successful postdoc with John Boothroyd, who's an absolute expert on everything toxoplasma related. For those of you who are familiar with the field, you will have heard this name over and over again. And he was recruited back to Switzerland and he decided, you know what, I'm going to start something completely new, Giardia Lamblia. Very few people had ever heard of this parasite. Very few people even had any idea what sort of cell biology this parasite presented and why was it interesting anyway. So Giardia had been a neglected model or eukaryote for many, many years. And Adrian, I must say, was one of the few people who actually saw the potential and exploited it. I joined this group in 2010. And in 2019, as Joel said, I started my own group at the University of Bern. So now everything Giardia has transitioned to my group and Adrian focuses on toxoplasma exclusively. Now, one of the reasons why I love Giardia is that it's a eukaryote presenting eccentric cell biology. And I do have a couple slides where I hope to be able to convey this message to you. But let's start with the concept of moonlighting. For those of you old enough to remember the TV series, perhaps you do. Moonlighting was a very successful TV series about people having odd jobs at odd hours of the day and the night. And the concept of moonlighting is actually really important in cell biology because it's become apparent that proteins do not have always single functions. They might have different functions, unrelated functions, depending on where they are found, either within the cell or outside the cell. And I moonlight too. So this is why this talk is divided into two parts. The first part is about the work that I do during the daytime, where my group and I investigate protein trafficking mechanisms and machinery in Giardia, and we'll come to part two towards the end of this presentation. So keep the notion of moonlighting in mind because it will show up also in a cell biological context. So the first thing I want to do is acknowledge the people who have actually helped me on this journey. That's my group. We are a small and tightly knit group. From the left to the right, you see Corinna Virdam. She's a senior research associate in the lab. That's me. That's Irina Ballmer, who very recently graduated, wearing her really fancy PhD hat. David Barmus and Jonas Rugenwold. They are two more recently appointed graduate students that have already contributed quite significantly to the work that we do. And I do want to acknowledge these three extraordinary graduate students whom I had the pleasure of co-supervising when I was still in Adrian's lab in Zurich, Jon Paulin Zuntor, Lenka Chernikova, and Rui Santos. Now, the work that I'll present today is actually work done by these people here. So what are the questions that drive us in the group? They're actually rather straightforward. The first question is, how does Giardia release factors that affect hosts? So Giardia is a parasite. It has to ensure that it survives and feeds within the host that it parasitizes. But how does it ensure that this happens? So what does that interface with the host look like? And how does it release factors that affect the host that's holding it? So what do these factors and what do the machineries associated to these factors tell us about mechanisms for protein trafficking and their evolution? How are Giardia's unique feeding organelles involved and how do they work? And can we reconstitute these mechanisms in vitro and how? So these four statements might look, or questions might look slightly disjointed, but I'll do my best to show you how they are, in fact, actually quite closely connected. So coming to our experimental model, I don't want to go into the details of Giardia's life cycle, but just to highlight what happens in vivo. Everything starts with a cyst. And the cyst is the infectious agent, but also the transmission agent for giardiasis, the illness that is caused by Giardia, also known as beaver fever, as I discovered when I hosted one of Joel's very talented students, Shweta Vipalia, a few years ago. Now, if if a host, which is basically any mammal, ingests a sufficient number of cysts, they will go through a process called excistation in the stomach. So the cysts will differentiate into the flagellated trophozoite form. That's the form that causes illness. That's the form that causes the diarrheal disease. It proliferates in the small intestine of a parasitized host, and that's where the malabsorption and malnutrition associated with diarrheal disease kick in. So a handful or a fraction of these flagellated trophozoites will differentiate back into cysts in a process called encystation. And this process requires secretion of very large amounts of material that allow for the encapsulation of the parasite into a very resistant, basically impermeable wall. And then the cyst can travel along the rest of the GI tract and be released with the diarrheal feces, and that cycle can start over and over again. Now, what really matters for this parasite are perhaps, at least in my opinion, 3 key points. The first one being encystation for survival. If a trophozoite does not find ways of turning back into a cyst, it will never survive outside a host. So it's really important for that cell to be able to properly secrete, and that's exocytosis. But the parasite is a parasite, it needs to feed. It needs to feed off of the nutrients that are contained within the host's gut. And feeding, we shall see in a moment, actually very much depends on endocytosis. And then the third part is virulence factor secretion. How does that happen? So that relates back to our question of how does Giardia release factors that might impact hosts? I don't have any time to go into the large amount of work that I did on the exocytosis story, but I will today focus on endocytosis and on virulence factor secretion. So I've often said Giardia to me is a really beautiful cell, and I'm a simple person and I love simple things, and Giardia is simple, apparently, in that it has a very reduced number of subcellular compartments. So what you see here are images taken at a fluorescent light microscope with specific markers for specific compartments of the Giardia cell. So in blue or cyan, you have the endoplasmic reticulum, which spans the entire volume of the cell. It's the largest organelle in the cell. You have the 2 nuclei. This is a diplomonad, so this cell has 2 nuclei, equivalent nuclei, so not like ciliates where you have a macro and micronucleus, but they're equivalent. You have mitosomes. So mitosomes are basically the low-cost version of mitochondria in a certain sense, so there's very little in them. They don't have a genome. In Giardia's case, they don't make any of their own protein. Everything they contain needs to be imported from the cytosol. And then we have very special organelles that are responsible for carrying the cyst wall material once the cell differentiates or starts its process of differentiation into a cyst. And these are called the ESVs. But for today, we're going to be focusing on these organelles in green here, which have been called peripheral vacuoles for several years and have now been recently renamed actually by us peripheral endocytic compartments. So, because the name is very long, I'll refer to these as PEGs from now on. So wherever you see PVs on certain images, please be aware these are PEGs. So they're the same thing, okay? Now, why are PEGs important? So this is work that started with Jan Paulien in Adrian's group when I was a postdoc in his group back in 2016. PEGs are important because it had been recognized since a while that they are responsible for whatever the parasite takes up. So you can run a very simple experiment where you take Giardia, which we can easily grow in the lab, we can cycle also through its different stages in the lab, so it's a good model in that sense. And if you feed it a fluorescent molecule that is metabolically inert, in this case dextran, Texas red, what you always invariably see is that the periphery of the cell lights up, and there's that tiny bubble of red in the middle that you see up there in the cell. Now, what's happening here, what compartments is this metabolically inert fluorophore ending up in? It's ending up in these little white bubbles, which I hope you can see, underlying the dorsal side of the parasite. So Giardia actually has polarity. That's also interesting about this cell. It has an apical side and it has a caudal side, but it also has a dorsal side and a ventral side. The ventral side, if you do a cross-section through a cell, the ventral side is what you see at the bottom, and that's where Giardia sticks to the mucosa of the small intestine and stays there. And on the dorsal side, it has an enrichment for these whitish looking compartments there. So this is an image taken by focused ion beam scanning electron microscopy, where you literally just go in and slice cells, and then you take an image, then you slice the cell, you laser deplete another layer, and then you take another image. And what we noticed when we went through these rather large data sets was that if you take individual organelles and you zoom in and try and see how they're located or how they're structured, what you almost invariably see is an invagination that seems to reach down to that organelle coming from what is presumably the plasma membrane, okay? So what you have is an organelle under the plasma membrane, and corresponding to that organelle, you have this invagination. So we were wondering about that. And so what we did is we reconstructed, using elastic, all of these images. And if this is the way we were looking at the cell, so the cross-section, now we're looking at it from the top, so we're looking at its back, at its dorsal side. And what we found was that if you reconstruct individual pecs, which at the time we still called PDs, and you reconstruct the presence of these plasma membrane-derived invaginations, what you find is actually a network of polymorphic organelles. So we're very far from this dogmatic view of, bubbles under a membrane, but we're looking at something that is quite ramified. And what's really interesting is the positioning of these plasma membrane-derived funnels. So what we think is happening here is that this is the way that Giardia takes up material from the extracellular space, because there is no other way into the Giardia cell if not through these organelles. There's no cytostome, there's no cytopharynx, there's no flagellar pocket, so these are structures that are found in other protist parasites that generally do a bulk uptake thing. With Giardia, everything needs to be taken up through these organelles, and apparently the way it works is the plasma membrane invaginates. There must be membrane fusion for that to happen. Remember that Dextra and Texas Red did enter those organelles, otherwise it would never have been inside. And that membrane fusion event then allows things to enter the organelle. Now, I might have to move this down.
Just click on OK.
Okay, good. So Hui then said, okay, I need to take this a step further. I need to understand whether or not this heterogeneity that we're seeing in the reconstructions is real, whether it's an artifact due to the way that the cells are prepared for the FIB-SEM process. And so what he did is he used a dextran derivative again and used stochastic optical reconstruction microscopy. And to cut a very long story short, which took several months of very hard work, he basically described 3 different categories of organelles, which he termed spherical, tubular, and polymorphic. And they're clearly distinct. They have a different surface area, but they also, as a result, have different volumes. So it's clear that this apparently homogeneous population of organelles is in fact not homogeneous at all. What this means in terms of function, we have no idea. So is it that we have different shapes that then correlate to different functions? Are we looking at intermediates of organelle division? So are the larger ones the ones that are just about to divide? This is all data that is acquired in fixed cells, because storm on living cells is still something that is very difficult to do because the procedure itself is very stressful to cells. So we are limited in that sense, but given the level of resolution that we enjoy here, we're fairly confident that these 3 subpopulations actually exist. What that means is still something that we have not understood. So in case any of you are interested in following up on the question of how these organelles fuse transiently with the plasma membrane but still regain their identity, Because this cannot be a stable process, right? It cannot be permanent. These things are special. The plasma membrane has a special composition. So how does that happen? If you are interested, please get in touch. This is a project that I shall be pursuing in collaboration with Joel here. And so I'm very keen to hear from you if you are interested in digging into the nature of how PECs maintain their membrane identity. Now, PECs are peripheral endocytic compartments, right? So it's time that perhaps we take a look at endocytosis. We're talking about organelles that are Giardia's feeding organelles. This is how this parasite takes up nutrients from the host's gut. And this is an uptake process, and generally uptake is associated with endocytosis. So Pauline went on back in 2016 and asked the question, well, what's happening with clathrin in this organism? You might know from Joel's really extensive work and the work of his group that membrane trafficking systems are wildly different across different lineages of eukaryotes. Some things are there in some lineages, some things have gone missing, some things have been reinvented, and so on and so forth. So the diversity of membrane trafficking systems is truly outstanding. And Giardia is no exception to that. So we asked the question, what's happening with clathrin at PECs? And very interestingly, Giardia's clathrin heavy chain, which is one of the two, the other one being the light chain in most systems, actually localizes to what are very likely to be PECs. So that was interesting, right? But the notion of them being endocytic organelles was, kind of supported by this observation. But what is really special about this clathrin heavy chain is that, for those of you who remember their basic cell biology or who are currently studying cell biology, this is a very strange clathrin heavy chain we're dealing with. So generally, from what we know from, the model organisms, basically a handful, clathrin assemblies are highly dynamic, they turn over very quickly, they tend to assemble in clathrin-coated vesicles, which means they do their job of picking stuff up from the plasma membrane and they travel to endosomes, Giardia's clathrin heavy chain is rather atypical. Basically, what we did was we tracked these individual particles corresponding to clathrin assemblies using a clathrin GFP reporter expressed in transgenic cells, and we asked the question of their longevity. Now, when there's high turnover and we have a very dynamic system, longevity is very short. But if you have a very long-lived structure, what this indicates is that that turnover is actually very scarce. And we realized that the vast majority of the events that we were imaging were actually very long-lived, meaning that unlike other systems where clathrin is highly dynamic, this clathrin in Giardia is not dynamic at all. And then the second question we asked was, okay, so this clathrin heavy chain is really weird, it's not dynamic, doesn't seem to turn over. What is it associated to? So can we reconstruct the interactome of this heavy chain and try and find perhaps elements that are known from other systems? So what is its entourage protein-wise? And so we took a line that's basically the clathrin heavy chain line, and we pulled down clathrin heavy chain. And what we were really surprised by was how streamlined this interactome is. So in yesterday's version of this presentation, I had a slide where I actually showed the complexity of endocytosis in a mammalian cell, and I had to take it out for reasons of time. But for those of you who might remember that, there's at least 50 proteins involved in that process. So clathrin heavy chain is associated to about 50 proteins. In Giardia, clathrin heavy chain is associated to a handful of proteins. Some of them we know from other systems, like we have a DRP, that's a dynamic related protein, that's fine. We've got AP2, so the adaptive protein complex 2, alpha, beta, mu, and sigma are also in there. We have a handful of lipid adapter proteins, but essentially this is just a fraction of what you would find in a full interactome. So it's got very little turnover or close to none, at least none that we could detect in reasonable experimental times, and it has a very streamlined interactome. So it's a rather atypical heavy chain. And with respect to the three morphologies of PECs that Hui identified, it was clear that there is a differing number of clathrin foci, clathrin assemblies, depending on the kind of organelle that you're looking at, which kind of makes sense because a larger a larger organelle might just have more landing spots for more clathrin assemblies. So if we need to, if we go on and try and summarize this, we have an atypical clathrin heavy chain. We have very little turnover. It's immobile, so it's not moving at all. It's very abundant. There's no evidence for clathrin-coated vesicles in Giardia, yet another aspect of the eccentric cell biology of this organism. And usually where you find a heavy chain, you generally find a light chain, right? And there's no bona fide light chain that can be identified in the interactome, not only, but also in the genome in general. So the statement would be at this point, or our conclusion would be that there's no identifiable clathrin light chain, or is there? So this is now one of these twists that that Joel and I, found extremely exciting, and it's part of the work that we're planning on doing together. So let me expand on this a little bit because I think this is important. We decided we'd take a closer look at this ORF here, 4259, the blue ball, because this is one of these proteins of unknown function specific to Giardia that we could find no match for in any database. So we asked ourselves, well, is there anything that we can do at least on a structural level to kind of figure out what it might be doing? The first question we asked, of course, was, is this true? Does this even localize to where we'd expect it to localize if this were truly associated to clathrin heavy chain? Then it does. You can see that the 4259 ORF and our heavy chain are pretty much sitting in the same place. And then what we realized, and this was work that we did in collaboration with Shweta Pipalia and Joel here at the University of Alberta, and Hui, who actually came over to train with them and had a great time doing so. What we realized is that this ORF of unknown function is predicted to fold structurally in a very similar fashion to bona fide light chains. So this is a comparison actually between, I forgot to label this. This is the Giardia one, and this is an overlay with the Trypanosoma brucei one, which is predicted to be a bona fide light chain. And here you have a handful of species showing you how there is a quantifiable and significant level of similarity between these structures. Now, this says nothing about its function. I want to underline this. Having a similar structure does not necessarily imply that you're going to have a function, and this is part of the work that we'll be doing in the coming months. That's exactly what I said. So what's the story with Giardia's clathrin system? This has actually turned into a very exciting graduate thesis spearheaded by Jonas and in very close collaboration with Joel and Frances Brodsky, who is basically the queen of everything clathrin, right? She has an amazing CV. She has outstanding experience. She knows about all the tools and the tricks and the tips when it comes to working with clathrin. She's based at University College London and is actually quite fascinated by Giardia's atypical clathrin complement. So I think we found a perfect match here with someone who comes from the mainstream, but who is very happy to be looking at something that is slightly off the radar. And in this scheme, what I'm trying to summarize is one of the approaches that we're taking experimentally, where we're asking the question of whether Giardia's analog to clathrin light chain, that's what we're calling it now. We don't know if it's an ortholog. Might be. Maybe it's diverged sequence-wise to the point that we cannot really recognize it. We don't know. We have no way of telling right now. So we're calling it an analog to clathrin light chain, ACLC. We're asking the question if it can functionally complement clathrin light chain knockdown mutants in ciliates, which have a diverged endocytic system but still maintain all of those pieces that are readily identifiable. Hence the rationale for choosing ciliates for this kind of work. Okay, so we've looked at feeding, which really matters if you're a parasite. I mean, it matters to everybody, but if you're a parasite, it matters a lot. And so we've looked at how these organelles, these pets, are involved in uptake, that are associated to a unique or rather atypical endocytic complement in terms of clathrin. So the other important thing that a parasite needs to do is survive within its host, right? So what about the ways in which Giardia releases information or sends information out to ensure its survival? This is an old slide, I know, but I think it summarizes things quite well. Giardia secretes lots of proteins. So Giardia secretomes, both exenic secretomes, but also co-cultivation secretomes, are ridiculously chock full of protein. And this is something that also has been seen with Trichomonas vaginalis. I just recently interacted with Abhishek, right, from your group, with Honza Takezi at Charles University in Prague, whose first project was about how many proteins Trichomonas vaginalis, another Procus parasite, releases in the extracellular medium. Which makes one wonder, right? Like, why are you sending out so much protein? What are you going to do with this information? How is it impacting whatever is sitting on the other side of the parasitic membrane? So Giardia secretes many proteins, but mostly we do not understand why it's secreting these proteins, what immediate functions these proteins should have. And when it comes to secretion, now we're going to go back into mainstream. Generally, when we think of secretion, most of us will think of this canonical ER to Golgi to plasma membrane pathway. So you start off with a secretory protein, perhaps a soluble one, that is produced in the ER, and that ultimately will find its way out. Let's say we're looking at, secreted in the so not literally from inside the cell to the outside of the cell, okay? Now, the thing about these secretomes is that they are chock full of proteins that have no features of conventional protein secretion. So I hope no one's going to sue me for using the UPS logo here. But when we think of UPS in the lab, we're not thinking UPS, the shipping company. We're thinking unconventional protein secretion. This is because these secretomes are full of proteins that have no predicted signal peptides, that show no hallmarks of transport through a proper secretory compartment. They have no transmembrane domains. They are things that are as banal as enolase, for example. So many of these are metabolic enzymes, right? Alcohol dehydrogenases, things like that, things that most of us might not be spending too much time on thinking about if they're focusing on secretory protein trafficking. And the thing about these mechanisms is that they are actually quite widespread. So I'll give you an example. Inflammatory responses in humans and most mammals are mediated by a complex called the inflammasome. The inflammasome is an ad hoc assembled complex that allows for the rapid release of cytokines. Cytokines that are released during inflammation are not released through a standard secretory pathway. They are released in bulk through this ad hoc complex. This is an example of unconventional protein secretion, where you have release of proteins that are never naturally engineered to actually be released. So if you were to look at them on a sequence level, you would not think this is a secretory protein. Now, when I started my group in 2019, I have done a lot of work on some form of canonical secretion in Giardia. And then I started asking questions that have to do more with virulence. And these secretomes had started coming out from several labs. And so I wondered, can we actually use Giardia as a simplified model to study unconventional secretory protein trafficking? And when you think about it, that might be a good idea because Giardia, for instance, lacks a Golgi apparatus, right? That's a fundamental secretory compartment that this parasite does not appear to have. It does not have proper endosomes the way we understand endosomes. So this is a very reduced subcellular compartmentalization that we have that we can actually exploit and harness to ask questions that have to do with secretion in the simplified eukaryotic cell. But perhaps most relevant to this point is the fact that there's evidence for unconventional protein secretion across many different species of parasites and the parasite-host interfaces associated to them. So this is where moonlighting comes back, right? We have all of these proteins that are not supposed to be secreted, they're being dumped outside cells. This is happening across multiple species. It's happening in Giardia, it's happening in Plasmodium, Trichomonas, Trypanosoma, Leishmania, and many of these are actually shed. So enolase was not mentioned by mistake. Enolase is one of those proteins that you will regularly find in the secretomes of all sorts of different species of cells. And there's lots of them, there's many more. These are just a handful that Irina decided she would try and track in terms of where they have been reported. So this might be an example of moonlighting for these proteins, because these proteins are essentially abundant metabolic enzymes in the vast majority of cases. But outside the cell, they might be doing something different. So we asked a very specific question: how are these proteins even released? right? They carry no secretory signals, yet they're found outside the cell. Is there a mechanism that we need to investigate to understand how that happens? And this is what Irena took up as a challenge. And amongst the many things that she did, she screened for several unconventionally secreted substrates or proteins that have been reported in the literature in these secretomes, and she found 4 that were actually associated to our old friends, the PECs. I think you can see that from the peripheral signal in green for our protein reporters, compared to a marker in red, which is cholera-toxic meat, which we use as a counterstain in these organisms. And when you do an actual, let's say, more robust analysis of signal overlap, what you clearly can see is that with respect to a platform heavy chain control, which is entirely PEX associated, and with respect to a cytosolic control, these 4 constructs appear to be somewhere in between. So there's a fraction of these proteins that is clearly associated to these organelles. So why would we have unconventionally secreted proteins associated to organelles that are known to be important for uptake, right? It's the wrong direction, if you will. We're looking at organelles that are doing this. Why do we have associated proteins that are then known to be released? This is a complicated scheme, but essentially all I want to tell you with this slide is that when we dug and mined into the interactomes of these specific proteins that are associated to PEX, we found very strikingly an enrichment for a family of proteins called alpha Giardians, which had been investigated to some extent about 2 decades ago and then had been abandoned, neglected. In fact, I knew so little about them, I was like, oh, they're probably not very interesting. And then someone in my lab, I think it was Irena, said, no, but you know what, maybe we should take a closer look at these things. And I'm like, oh, okay, knock yourself out. Well, not literally, but anyway. What are alpha Giardians? Alpha Giardians are human annexin homologs. I don't know if any of you are familiar with annexins. They have been studied in a lot of detail because they play very important roles in all sorts of things. They can modify, they can induce membrane curvature. They're important for clotting. They're part of complement. They do all sorts of things in humans specifically. But what's interesting to us is that they can bind to or insert into the plasma membrane via negatively charged phospholipids. And even more interesting, some annexins have been reported to be secreted unconventionally. So that was neat. I was glad that Irena had told me to take a closer look. This is a very simple alignment to show you that annexins are very easily recognizable because they consist of repeats of a specific annexin fold. So it's quite straightforward to actually find them. So those are the alpha Giardians, and alpha Giardians are annexins, but what do annexins do in more detail? They basically can disrupt membrane permeability in a calcium-dependent fashion. So they're able to bind lipids, they have this core that is built from the 4 repeats that I just mentioned, and they can destabilize membranes. They can interfere with membrane integrity. So we asked ourselves, is there a link between unconventional protein secretion and PEX that is mediated by Giardians, alpha Giardians as annexins? So here we have an organelle that has not fused to the plasma membrane. It's not ready to take up. We know that the complex that we've identified that includes these UPF substrates is sitting at the PEC membrane. Is this happening then? So when the PEC fuses to the plasma membrane, because of this complex that is enriched for alpha Giardians, are we then having a slow release of things that permeate across the membrane because the alpha Giardians are destabilizing the membrane and letting this leaching phenomenon happen with small amounts of abundant cytosolic proteins that just happen to be coating the surface of these organelles. So in other words, are PECs not only capable of taking up, but are they also capable of mediating release unconventionally? So there are 3 predictions that can be made to test this model that we have here. So you see this very detailed image here of the complex that we've identified and how we think things might be working. So there are 3 predictions that we can test. The first one being that alpha Giardians, much like annexins, will be able to bind lipid membranes. The second prediction is that alpha Giardians can revert membranes, so destabilize them. And the third prediction, which is in italics because I have no data to show you for this prediction today, is that not only can they traverse membranes, but they can actually drag proteins with them. So let's go for prediction one. This is where we test the ability of alpha Giardians at PEX to bind lipids. So when we looked at all 21 alpha Giardians, because there's 21 of them, we have focused on only 2 so far, we found out that there were a few more at PEX, many of them cytosolic, but a handful at PEX. And we used lipid strips to interrogate their ability to bind lipids. And what we found was that of the alpha Giardians that bind PEX, those are capable of binding negatively charged phospholipids. And when you do this in a little bit more detail, where you interrogate a preference for PIPs, I'll come to PIPs in a moment, what you find is also some degree of promiscuity, but a certain preference for specific PIPs. Now you might be thinking, what are PIPs and where to find them? This relates to work done by Lenka Chernikova back in 2019. Things have a way of coming back from time to time. So what are PIPs? PIP stands for phosphatidyl inositol phosphate. So PIPs are very specific lipid residues that are present at very low abundance in cells, and they mark specific membranes for specific things. So they generally mark sites of membrane fusion and reorganization. They're organized in patches. And what's really interesting to us is that Lenka, in her graduate thesis, demonstrated that PEGs are actually decorated with PIPs, okay? So we have a situation where we have an organelle. This organelle carries a complex that might be involved in letting membrane proteins through. And the way this complex might be anchored there is by the binding mediated by the alpha Giardians to specific PIP residues that we already know experimentally are present on this organelle, as shown in this scheme coming from Lenka's earlier work. So this got us very excited because it allowed us to ask the question, can we mess with this? So we have these alpha Giardians. We're using alpha-1 Giardian as a, let's say, poster child for the family. We have these proteins. We know they're associating to the organelles, but can we mess with them? Can we interfere with that? In other words, can we determine what is important residue-wise for their binding and localization? And we generated a handful of mutants. We're generating more. And with respect to alpha-1 Giardian wild type, which is the first quadrant that you see in green, an N-terminally tagged version loses its ability to associate to these organelles, so the N-terminus is important. A phosphonull version, a predicted phosphonull version, doesn't know that it has to go to organelles, so that seems to be important. And interestingly, a mutant that was not meant to bind lipids anymore still happily associates to these organelles. So it's going to be more complicated than us just going in and changing residues by site-directed mutagenesis, but we have some insight in that also from their lipid binding behavior. So our G25, which is our wild-type alpha-1 Giardian, is our reference. The one right next to it, the V5 tagged version, does not go to the organelles and kind of loses its ability to bind lipids compared to the wild type. Same holds true for the phosphonull mutant. Loses localization and doesn't bind, and our binding mutant actually seems to be doing pretty much okay. So there are more mutants coming into the pipeline, which I have no time to talk to you about, but as I was mentioning to one of the students during our pizza lunch, we ask questions that have to do with interactomes, and we're interested in getting the best interactomics data that we can possibly get. So we started off doing our IPs more or less in standard conditions, and now we've moved into something called XLMS post-capture for structural information, which I do not have time to talk to you about in detail, but this is just to say that with this technique, we can not only acquire information on the composition of a protein complex that is isolated, but also on the relative configuration of the protein components. So I'll skip that. Prediction number 2, can they traverse membranes? We tested this with liposomes. The long story, the short story of the long story is that they do destabilize liposomes to some extent. So what you see here is an assay where you have a liposome that contains high amounts of a dye that self-quenches at high concentrations. But when there's membrane destabilization, some of that dye can exit, and this causes fluorescence. So we have an increase in fluorescence only in the presence of alpha-1 Giardia, meaning that we're having some degree of membrane destabilization. It's not huge, and maybe it's because it's only alpha-1 Giardia on its own, but it's there. So something is happening, at least in vitro. So our focus is really on PECs at the moment, on three aspects of PECs. So PECs as release platforms with the whole alpha-Giardia story, the PEC-PM relations and resolution. This is where I'm looking for someone who feels like being challenged by the Giardia system. And also Jonas's project PECs as uptake platforms in collaboration with Joel and Francis, trying to figure out what the evolutionary history of clathrin is. Now, because I moonlight and I have like 3 minutes, I'm going to have to.
You got more than that.
Well, I have more than that. Okay. So, as I said, I moonlight, and part one was dedicated to what I enjoy doing with my group members, and I hope they have as much fun doing that as I do. And part two is all about co-directing the Multidisciplinary Center for Infectious Diseases, which is the newest strategic center at the University of Bern. So Uni Bern likes to basically maintain strategic centers that act as interdisciplinary research platforms, connecting faculties where people from different walks of life come together to try and do something different. And this was the understanding behind the origin of the NCID. We launched operations in January, February 2021, and our mandate is to promote the study and mitigation of healthcare, societal, ethical, and economic risks from infectious diseases. And I think here you'll see why we're called multidisciplinary for infectious diseases as opposed to the Medical Center for Infectious Diseases. We represent a total of 7 disciplinary clusters. So this is where these are the clusters that our 79 members are distributed across, from immunity to neglected diseases, economics, microbiology, patient-focused research, epidemiology, and society and law. And at the center of all of this are our 3 core activities. Core activities are intended both as service, but also research-oriented platforms. One of them being the Be Ready cohort for a peacetime cohort of the Bernese population, one of the things that was sorely missed in pre-COVID-19 times. The Biopreparedness Biobank for high consequence pathogens, so that's a BSL-3 level, and Ethics and Policy Lab, which is actually quite unique because this is where policy and ethicists, experts can actually help people like me, when they show up with data that they think is relevant on a societal level, they help me translate this into something that might be of impact that can be implemented. So we're composed of 7 clusters. We fund a total of 23 research projects, which is why I'd like to focus for a moment on our research efforts. So we launched our first funding call in 2021, and the projects are just about to end, actually, for a total funding volume of 8.4 million francs, distributed across these 3 themes. So these 3 themes constitute the remit of our funding call, the investigation of infectious threats, the development of surveillance and preparedness strategies, and solutions to manage and address the impact of infectious diseases on animal and human life. And I have no time to talk about our teaching efforts, which are actually quite substantial, but I'd like to focus on what Joel has mentioned. So one of our outreach activities has to do with pushing the boundaries of the NCID from being not just a national center of competence and reference for local authorities, but also having an international scope to its activities. And thanks to our involvement with the Guild, which is a university alliance that's very active on an EU level, we were able to secure leadership in collaboration with Stellenbosch University in South Africa and the University of Tübingen in Germany of a cluster of research excellence dedicated to genomics for health in Africa. So what you see here is a map of where all of our consortium partners are based: Ghana, Rwanda, Nairobi, and so on and so forth. So the largest fraction of our group is really based in Africa, and we have a handful of very, very active, very valuable partners in Europe. The idea behind Genomics for Health in Africa is to leverage the potential of genomics to revolutionize healthcare in Africa. And the way we're going about this is we're taking genomics and we want to apply them to 2 very different lines, let's say. One that has to do with rare diseases and familial cancers. There are plenty of patients in the African continent with a rare disease or with a cancer that have no hope of a diagnosis because there's simply no resources to be doing that properly. So this is something that is really revolutionizing healthcare now, thanks to the efforts of one of our colleagues, Shahida Musa, based at Stellenbosch University, one of, I think, of like 10 clinical geneticists on the continent. So this gives you an idea of how difficult the lives of these patients are. And on the other hand, of course, we have the whole genomics as a tool for surveillance when it comes to infectious diseases, pathogens, and their hosts. So we've combined this with a group of colleagues across Africa and Europe who are interested in the structural biology side of all of this. So once you have a sequence, what are you going to do with that sequence? You're going to try to understand how this translates into a protein that can be modified, manipulated, utilized for something that's more than just information. And I believe that's it. There would have been so much more I would have wanted to tell you about the MCID, but I guess you're just going to have to invite me another time. And if you do want to stay updated on what we're doing, I'm also very happy to share these slides, by the way, if this is of interest to anybody. Because there are hyperlinks in here, just makes life easier. So if anyone is interested in staying updated on what we do, we have a regular multidisciplinary seminar series that's also available online. We have a newsletter, which I think we very aptly call The Spread. It's dedicated to all of our activities. There are interviews in there. We have a very special focus on researchers. That's very important to us, the next generation of researchers who will undoubtedly be facing more infectious diseases threats. And of course, the Genomics for Health in Africa initiative, which I'm very happy to talk more about perhaps later on. With this, I'd like to thank you all for being so patient and being very, very generous with the extra time that you've given me. I'd like to especially thank Joel for inviting me. Our funding bodies are, of course, very gratefully acknowledged. And last but not least, let me give out a shout out to ICAP. ICAP is the International Conference on Anaerobic Protists. This is something of it's a passion project in a certain sense. It's rather niche, but it's so interesting to meet colleagues from the Trichomonas, Giardia, and Amoeba, Cryptosporidium field. There's a lot of molecular parasitology done there, but also a lot of epidemiology and taxonomy. So if you're interested, please join us in Auckland. This year is a little far, but it's up to you. You can turn it into a holiday. And thank you very much for your attention.